Anyway, I want to welcome all of you to the second lecture in the Green Family Lecture Series for 2016. I'm Russ Kaplich, I'm the director of IPAM. I'll say a few things first about IPAM and about this lecture series. Many of you were here yesterday, so this is partly a repeat. I think I'll say it in a different order so it sounds good. IPAM's mission, well, the statement of our mission is that we foster the interaction of math, both pure and applied, with science and technology. Obviously, that's a little off now with this group. So I would say we have expanded our mission to include interactions with other fields, and I would say with any discipline, but this group certainly extends that stated mission to humanities and arts, and probably other fields I'm not quite capturing. We also promote mathematical innovation, and we develop, one thing we really hope from, and especially it's true for this group that's in the current program, we are trying to create new research communities by bringing together people from different fields to a common ground here at IPAM. And we hope to show the power of math and to have math in those research communities. And our aim is to transform the world through mathematics. Our funding is primarily from the National Science Foundation. We just got renewed last year, which is a good thing for us. We are also in the midst of a transition. We're looking for a new director. I expect to step down as director in a year after 9 joyous years of doing this. And that's not sarcastic. I mean it. It sounded sarcastic. I don't say that. We're currently undergoing a 15th anniversary campaign of fundraising for IPAM, and so I would encourage you to engage with that. We would certainly love to have your support. The Green Family Lectures that this is part of, I think this is our 5th year, and we've had a stellar list of speakers. This lecture series is endowed by a generous donation from the Green family, and Mark Green here, who was responsible for this gift, is one of IPAM's founders and first directors, and I would say has been the absolute leader of IPAM, and really in establishing this institution and the great tenor of bringing together people from different disciplines. We heard a wonderful talk yesterday from Ingrid, and where I think you heard the unique combination of depth of mathematics and exciting applications. But I'm actually going to leave it to Andrea Bertozzi to introduce her tonight.
Thank you, Russ. Okay, so this is really an honor to introduce Ingrid Daubechies for the second Green lecture. So I'm going to be a little less terse than I understand Stan was yesterday, because not everyone may know all of her honors. And she tells me she embarrasses easily, so Ingrid, please don't be embarrassed. So I'm going to really embarrass you because I understand you're a baroness now. Oh. So I sound like a idiot, sorry. But I will. What I do want to say though is Ingrid actually is such a wonderful role model to so many people, and especially to women in mathematics who are still an underrepresented group. And for people my age, I'm 50. I just turned 50. God forbid. She's one of the few that people from my generation have as a role model. And in 1994, when she was that the right year when she moved to Princeton and became the first female full professor in that department. I'm a Princeton alum. I did all my degrees there, and the entire time I was there were never any women faculty. And you sort of start to wonder if maybe it's never going to happen. Like it's just not possible. So when Ingrid moved to Princeton, I thought, okay, thank you, Lord, for making this happen, right? And she left Princeton, I guess a while back, in 2011, right?
Yep.
To move to Duke, where she is now the J.D. Duke Professor of Mathematics and Electrical Engineering. She has so many honors, I can't name them all, but they include a MacArthur Fellowship, National Academy member, American Academy member, the AMS Steele Prize, and many other awards. I also wanted to share one little anecdotal story. When you're thinking back to all the sort of special moments in your career when you were young, you sort of make a small short list of things that people said to you, or moments where you learned something. And there's one event that I remember very, very well, which was one lunch that I spent with Ingrid at Bell Labs back in the 1980s when I was a graduate student. And she didn't know me at all. And I had been warned by the senior people at Bell Labs that Ingrid was an amazing scientist, right? And so she pulled me aside at lunch one day and just taught me wavelets. I mean, I got a full lecture from her, written out on about 8 pages, handwritten, in absolutely beautiful penmanship that I will never have. You could see the chicken scratches here. If we had the technology today that we had back then, I would have taken out my phone camera and taken pictures of those notes. But unfortunately, I didn't, and I lose pieces of paper, so I don't have those. But at any rate, she's been an incredible role model to me and probably to countless other people, both male and female, in our field. So without further ado, I'd like to introduce Ingrid's next talk, which is Bones, Teeth, and Animation. Thank you.
Well, thank you, Andrea, and you did make me blush. This talk will be very different from the one yesterday, although it's a topic on which I've been working for a number of years and on which I'm very excitedly still working. There are a number of collaborators here, very interesting. I mean, I have an Iraqi young woman and an Israeli mathematician. I have 2 biologists, only one of whom I have a picture, Doug Boyer. I have Roy Perrin was a student of Yaron's, and then a number of graduate students. In particular, Ting Ran Gao, who is the upper one on the left, who actually, of whom I've stolen many slides for this talk. In fact, in February, I gave here at a different workshop a talk together with Ting Ran on machine learning, fiber bundles, and biological morphology, for which we had prepared slides together. And I wanted to be upfront for those of you who were there, but even of those of you who aren't and who look at it, I stole those slides from him. Okay, but animation. So the mathematics on which many of this is built come from computer graphics, and in particular animation. So let me tell you a little bit. That's the only thing about animation I'm going to tell you, because research itself is about biological morphology. But so animation relies on computer graphics, and computer graphics uses 3-dimensional mesh models for their surfaces. So every one of those rendered persons that you see or animals that you see is really a surface that gets animated and given color and texture. So the very first object of that type that was a real object was the teapot, because before that computer graphics people made other things. They made tori and two-handled tori and things, and then oops, I don't want the network. And the spouse of one of them said, "Why don't you take an object like this?" And she held up Melita teapot, this teapot, and so that was the teapot. So the teapot was the first, here I don't have it, has a few hundred triangles. It later became, this is a much more modern rendered teapot. And in fact, it's a kind of in-joke. In every Pixar movie, the teapot is somewhere. I mean, it can be sitting in a doll house and so on, but the teapot is there. So they became much more sophisticated over the years. Here are a few stupid little examples. They're a few years old already, but some fanciful architecture in a cartoon or a manufactured element in CAD. Here are 2 pictures from Pixar movies that actually show the change that happened in animation, because on the left, this is from Toy Story 1, Woody is all in NURBS. I mean, piecewise splines. And on the right, a character that first was Jerry in a little short and then became the puppet doctor in Toy Story 2, is made with subdivision surfaces. Subdivision surfaces are much more versatile for animation because you don't have complicated stitching ways of putting surfaces together. You just have rules for going from coarse to fine, and you can change those rules and you can adapt for ridges and so on. And so a whole hand of Jerry is just one surface. Well, while every nail would have been a different nerve before. So now that was all. Animation is about making things and making them move. When they're humanoid, well, we'll come back to that. But I'm interested not in making objects, but taking objects and using the computer graphics tools to study them. So you can, of course, we know that, scan existing objects. You can do it with barcodes or with LiDAR or with yet other techniques, and we all know about Kinect now and how well that does. So when you have registered all these points, what you have is a whole point clouds that you can triangulate. You have to denoise it and triangulate. And you can, and here is an object, I mean, the original object, the virtual one on the screen, and then a milled one. I mean, these days we would 3D print it more often, but you can edit them if you have, and that's where animation comes in. You can, this was a subdivision surface reduced to a coarser level, and then some points moved and then refined again, and that makes the camel talk. But when you talk about doing science with them, you have different problems. If you have the same object scanned twice, how are you going to recognize that it's the same object? I mean, the points, all the coordinates are different. It's not just a question of translation and rotation. Even in the same position, you're not having picked exactly the same point. So, or if it's not quite the same object, how are you going to recognize that one looks very much like the other? And how are you going to quantify that? And a little playful example, here you have twice the same object, once big and once small, except that if you were to scale down the big one, then you would see it's not the same. I mean, it's not just that it doesn't have the same detail, it's also that if you scale down a Volkswagen to a dinky toy scale, it doesn't look right. It looks too long. So the dinky toes actually are more stubby. I mean, so, but how would you find that? I mean, these are questions that you have to think about when you, even in animation, when they animate a humanoid character, what they do, and you've all seen this, is they have actors on whom they put sensors, and they make the actors go through the motion, and those sensors will then give them the coordinates that will make it possible to make the character move and follow that motion. But that is something very interesting for people who want to use this computer graphics knowledge and insights to study locomotion of animals. I mean, at Duke, we have a lemur center. They started breeding lemurs when it was still legal to export lemurs from Madagascar. It's one of the biggest lemur centers in the world. They have many generations of lemur, and they are they are willing partners in many scientific studies. Cognition studies, for instance, I mean, because they're primates, they're curious, they love figuring out and getting little rewards and seeing how they can solve a puzzle and so on. They also like to, as I said, study locomotion, because these are threat species and because their habitat is changing. There might be areas where they have to move more over the ground instead of from tree to tree, some nutrition trees are no longer available and so on. So people want to know how much calories they need to spend for different types of locomotion and what they get out of their food. And in the rainforest, you can't do experiments like that. But if you have a lemur colony with lemurs that love to play with you, I mean, then you can. And so for the locomotion experiments, they do the same thing. They actually attach little sensors to these lemurs. And that's one thing the lemurs do not like. I mean, they do not like that they have to have a little piece of skin shaved and so on. So they very much would like to do experiments like these without having to find the reference points. And if one can do it for the lemurs, then it would be nice also for big budget movies, although they don't have the same financial constraints. But so in summary, we would like, when 2-point clouds correspond to the same surface or very similar surface, to recognize that, quantify when they're different, how much they're different, and find, if we have similar surfaces, points that correspond. And why do we want to do that? Well, my involvement with these problems all started with a conversation with Jukka Jörnvall. And I met him at a social event. And we started talking about what we do and so on. And I learned that he's a biological morphologist. He studies especially teeth and bones from extant animals and extinct animals. And his Doug Boyer was then his postdoc. He's now completely serendipitously, he became serendipitously, he became a faculty member at Duke, and so we're now at the same place. And he was at Stony Brook, and I was at Princeton when we started working on this. But so the very first project we did with Jukka and Doug, just as Yaron was joining my group in Princeton, and Yaron has this big background in computer graphics, but on the mathematical side of computer graphics, which I really liked. Many computer graphics algorithms are very ad hoc, but he really uses differential geometry to guide how he builds his algorithms, and I like that. So just as he was joining, I had met Jukka Jörnvall, and he had told me that he had a recent big breakthrough on finding some universal characteristics in the teeth of mammals. So he had found that if he looked at the molars of mammals and he looked at their complexity, the complexity of the chewing surface, that he could recognize from that whether they were insectivores, omnivores, herbivores, carnivores, and so on. And of course, we all know that the dentition of animals, and of course, teeth experts know that even better than we, but it was to a level that people had not expected, especially on the molars. And it was not a question of wear and tear, because this complexity that he could find was, he could use it for mice and for elephants. So he would first reduce it to one unique scale and then look at complexity. I said, how do you characterize complexity? And he said, well, he says, we are going to the digital world. Now we have these digital representations of these surfaces, and we look at them on screen. And when we look at them on screen, there is this rendering program, which is a computer graphics program that puts a red, a blue, and a white light, so that the reflection of the shiny surface actually gives you impressions as you look at that surface that really make your brain reconstruct the 3D information. Especially if you turn it a little bit, it's very good at that. And he said, so we do that, we take this rendering program and we turn the tooth towards with the chewing surface in the screen, and we take a snapshot, and I thought, and he says we look at all the faces, we see the blue and the white and the red, and we count the number of transitions. And I said, but what you're really doing is you're looking at changes in the angle of the normal on the surface. That is something that mathematicians have better ways of calculating. He says, really? So that was actually a kind of off-the-shelf thing. It was just looking at changes, at curvature. We were integrating local curvature, surface curvature over the whole chewing surface, and we did that. And Yaron wrote a little program with them and so on, and they were very happy about that because it was more accurate than their manual counting of these, which is not surprising. And so they called it the Dirichlet normal energy, and it's the DNE rather than the DNA. They like the acronym also. And that's now, if when they go to their specialist meetings, that's now one of the subsections of people who study teeth. They always have a whole side program on DNE and new things that have been found with that. But so that was a kind of off-the-shelf thing. But then once we could do that, it's always the same in these interdisciplinary. Once you can do something, they say, oh, maybe you can help with this. And so then we started working on what's really a much, much, much bigger problem, and on which we did not have, and still do not really have, an off-the-shelf answer. So he says, we'd love to have an automatic way of finding distances between surfaces. So of course they already try to quantify surfaces. So if they have a whole collection of teeth, then these morphologists, they look at the collection and they want to study this collection and characteristics and correlate it with other things that they know about the animal. They have enormous domain knowledge. They will look at this whole collection and they will register things that are present in the whole collection. They say, oh, this particular entoconid, or this particular, and so on. So they have names for all these things, which I have not learned. And they mark them then, they make a list, and then they mark them on each of these specimens. It used to be that they had very nice apparatuses with a stylus and so on that would register coordinates. Now they do it digitally on a screen. I mean, they turn the thing and they put a point and so on. So what they get is that they have a proxy. They have these landmarks on each of them, and then they use these n-tuples of points as a proxy for the surface. And to find the distance between the surfaces, what they do is they take the landmarks on one, they put them in as close a relation to the other as they can by rigid rotation, translation and rotation, and then they look at the sum of the squares of the distances. And that gives them a distance squared between that they call the Procrustes distance. And so they do lots of things with this Procrustes distance, but you see, it's very painstaking. And as they are putting more and more of their data online, I mean, people are scanning these surfaces and making them available online, it would be wonderful if it were possible to compare them, to find biologically meaningful distances between them without having to do this landmarking. Because the landmarking is something for which they need enormous domain knowledge, and so it can only be done by people who already know a lot about these data sets. They'd like this to be available to everybody who's interested in investigating these morphological surfaces of different animals. So they wanted us to find ways of finding distances automatically. And so we will have these finely discretized triangulated surfaces, and we'll try to find distances from them. Now, I had a whole talk that explained how we did things with those and what we did and so on, which I'll summarize in a few slides here because I want to go further than that and tell you about more recent stuff. So we actually came up with two different distances, which both worked pretty well. The first distance was one in which we first conformal flattened the two surfaces. Then for each of the two surfaces in the conformal flattening, the original geometry was still represented by the conformal factor. And so we could look at these two conformal factors and do an optimal transport of one to the other and construct it in such a way that it was conformally invariant. So not just a straightforward optimal transport. You had to have conformal invariant, but we did that. And another one that we did was by saying, look, and this second one we knew was going to be accurate, especially for when the distances were small. What we said is the way they put these landmarks down, yes, it has a lot of domain knowledge, but if you look at these teeth as well, the points are not exactly evenly distributed, but you could imagine a few more in between and have a fairly evenly distributed number of points on these surfaces, and then you would have maybe had 100 instead of 20. If they are kind of evenly distributed, then what that means is that each of them, if you look at its Voronoi cell, would have about the same area as the others. And so you could say to a first approximation that the mapping of these landmarks and so on is like an area preserving map. It's not really true, but let's think of it in first approximation as an area preserving map. Well, we can maybe think of putting ourselves this 200 or maybe 500 points at random, but well distributed on a surface, on each of the teeth, and try to find a way of mapping them to each other. And we found a fast algorithm for doing that, and that was our continuous Procrustes distance. You can define it in the set of area preserving maps. In reality, again, we did it via conformal flattening, and we found a conformal map. And there we actually proved that if the distance is small, that the area preserving map is very close to conformal. And so we could look the conformal one and then modify it to be a close by area preserving, and we would have something that was near optimal. And so we built those two distances. We built the algorithms for doing that, and we had prior to doing the whole study, agreed with the biologists what we would use as criterion for saying, yes, this is successful, or no, this isn't. And one way of doing it was to look at the distances as they would determine them with observer-determined landmark-placed distances. And here with our conformal Wasserstein neighborhood distance or our continuous Procrustes distance. And we would rank them so that nearby labels would be things with small distances. And we would then we'd compare the distance matrix for one with the distance matrix of the other. And we did that for both our distances, and as you see here enlargement, you see we do capture fairly well the fine structure of these distance matrices. Not perfectly, but fairly well. We had another test which was based on the distances we determined. Would we, if we tried to classify into genera and species, find the same thing? How successful would we be? And we had a very high success rate. And there was yet another test. What if we put landmarks and we transport them through our... Because what we're doing is really building a transport map in both situations, actually. So we transport them and we compare with the correct landmarks. How far are we off? What's the Procrustes distance between what we find and what they put? And so with all these criteria, we were doing very well and we were very happy. And we wrote a PNAS paper about it. And what we found actually was, this was even more surprising to me. If we went from this Microcebus to Lepilemur tooth, and we transported the landmarks, then we ended up with transporting this point to that one, while if we had done the transport on smaller hops, via an extinct common ancestor, or via closer related but still extant cousins in 3 small hops, then it would have been clear that this point really had to be mapped to that one. So here and not there. And in fact, this was suggested to us by Doug Boyer because it had been a recent controversy in morphology. There were experts who had put those points in correspondence with each other, and others who had pointed out, no, I mean, this is not the right thing, this was not your species, so you actually got it wrong, and had proposed. And so our algorithms made the same mistakes as a morphologist, but who didn't have a fine enough sample on the collection of species could have made. Okay, so that's where we were maybe 4 years ago. And but interestingly, things changed, and actually about 2 years ago, we started not making progress anymore. I mean, I knew, and I had told them from the start that this would be something in which we would do things and learn more and gradually make more progress. I mean, I did not expect with the D&E thing, we had gotten a tool and that was it, and it was software that we made available and people started using it. We said, this problem is more ill-defined, we need to learn more. You cannot expect that we will be there, it will take several steps. But that was a process that had gone very well until about 2 years it stopped, and it took me almost a year to realize what was going on. What happened was that whenever we discussed, they would always come back on points that I thought we'd already discussed before. And what really was happening was that, and once I articulated it, they agreed. The fact is that nobody articulated it. I mean, it took a year. What really happened was that they had seen our maps, and these maps were, in fact, it turns out, and we now all agreed, are more fundamental concept than these distances. These distances was what they had when they built a proxy and so on. It was a tool to get, but given that we were constructing maps as intermediate state, they liked those maps. And if something was wrong with the map, even if our distances turned out to be okay, they were not happy. I mean, the map was wrong. The map did not have the right properties. And so, but as soon as I realized that that was the problem, that shifted the whole point of view. I mean, and now we actually, and I'll come back to that at the very end, I'm at a point of view where I think we should really build mappings and use those as a basis for machine learning. And that's what we're building on now. But even so, even before we were there, Tingran Gao, the student on whose work the further slides will go, used this for very interesting insight. The other thing is that it's very important to them that there is no holonomy in. So meaning, you see what happens here is that if you start from one place and then regardless of how you get to the other place, you should get the same result. Because the reason for that is that they believe in evolution. And certainly, I mean, all these lemurs come from common ancestors. And so the correspondence between this species and that can both be brought back to a correspondence in their ancestor and coming down again. And so it shouldn't matter whether you go from A to B, B to C, or from A to C. Since it all comes from the same tree, it should give the same thing. So that's a very strong point that we only gradually understood. So Tingran Gao reformulated the whole problem as a very nice mathematical problem, a connection on a fiber bundle and horizontal diffusion. So let me tell you a little bit about that. But even before that, the very first thing he did was he did something very nice, which already was a little bit like a diffusion geometry about which I'll talk. So what he did was he said, if we look at all these mappings, then what we can do is we can make a very large concatenated matrix about distances. We have distances on the tooth and distances between teeth and so on. We can make an enormous matrix of all these distances. And if there really is a correspondence thing, a real correspondence thing, then it should be the case that points that are in correspondence of each other for the right matrix that you construct give you just an eigenvector. I mean, this is an eigenvector. This, and so on. So you should have a whole lot of eigenvectors with eigenvalue 1, which, I mean, if you make diffusion from one surface to the other costing very little and diffusion on the surface costing more, then you should have all these eigenvectors. So what he did was he built that matrix. He didn't have the true correspondences. And then he looked at whether it was possible to find eigenvectors that, for which things that had were very close to an eigenvector and that were very sparse. The idea being, if you have points that are in correspondence with each other, they will be sparse. I mean, the matrix is so noisy that real eigenvectors are going to be hard to give you true information on the... But if you input sparsity... And so what he found when he did that was already that, I mean, so the space is something consisting of these 4 surfaces. There's absolutely no reason, if I ask an eigenvector close to eigenvalue 1, for it to give me entries on one entry on each surface. It could choose to put all the entries on one surface. But in fact, what he found was that he found entries on these different surfaces. So you start seeing the correspondences coming out of the whole collection. So the mappings, this is with mappings that the biologists were not happy with. I mean, one mapping in one pair was not a good mapping necessarily. But if you had a collection of mappings, because errors would pull you in every so which way, they would give you much more. So this made us realize that just like the biologists themselves can do much more when they have a whole collection, we can do more if we have a whole lot of pairs of these maps. And so that's what brought us to bring the collection. So we want to use the information in the maps. And so in the continuous procrustes distance, what we do is we take all the points on one surface, and we want them to compare we want to compare them with points on another surface under some correspondence map that we have to find. So what you do is you take a correspondence map, which we choose area preserving. So you map points from here to there with an area preserving map, and then you take the points and you try to move them as close as possible to their transformed points, and you look at that distance squared. So that's the continuous procrustes distance squared, and I should have brought, I forgot, I actually printed these two in color in 3D printing, and so in February I brought them and they could go through the room, but well, you'll have to ask your friends who were here in February. And learning from distances. So we have all these distances between the individual surfaces. And if you use those to cluster, you do find that here I have 10 individuals of 5 different species, and the species are given by color, and you do find a cluster if you take a three-dimensional representation of that distance matrix more together. But if you do something better, that was just pairwise distances. If you now take these pairwise distances as something that you use in studying that graph via diffusion techniques, then what you can do is you can look at the diffusion distance, and I'll explain a little more in the next few graphs, but you see how that immediately gives you, with the same data, but taking them not as pairwise distances, but as something from which you're going to compute something on that whole set, as a first approximation for better distances, how you can get much better. And so you have these two distances here. So how did we get there? I mean, how do diffusion maps work? Diffusion maps, the idea is that you have a whole lot of points, and every point here represents one tooth on the manifold of all these teeth. And what you expect is that small distances are much more trustworthy than large distances. So what that means is that you believe that locally you know reasonably well what your manifold looks like. Locally, the distances that you measure, which are only approximations, but they're probably approximations that are fairly well for the tangent plane locally, and so they are locally good approximations for the neighborhood in which you live. Locally, you have distances in each of these neighborhoods, and what you can then do is look at paths in each neighborhood, and you can connect them together. And so you can find things that are far apart by going over the path that connects them. And that basically is what you do. You have distances that you do believe, you build a diffusion matrix with them, you then look at powers of that diffusion matrix in order to diffuse over longer distances. And I always, I visualize this as having a complicated piece of modern furniture and covering it with little doilies in crochet. I mean, and that will follow the whole thing. And I like to use craft metaphors rather than sports metaphors. So you do that, so you find a random walk on your graph, and you find its eigenvectors, and you use these then, and the expression, the values of those eigenvectors in each of the points in order to give a parameter parameterization of your manifold, and then you can build a diffusion distance using that parameterization. It's just an L2 distance in terms of these coordinates that you introduced. And that gives you that much better clustering. But, so that's what we found. In fact, and that's the thesis, that's part of the result of Tingran's thesis, you could then go beyond that diffusion distance. And he built, by looking at the whole thing as a fiber bundle, he built a distance that gave us this clustering. So again, the same data, but not just the distance matrix. Also using these maps. I mean, the maps that the biologists were, and so on. We have the maps, why not use them? And that's what so let me tell you what he did there. So, okay, so we have these correspondence between the surfaces. Now, that means that we have correspondences between our triangular meshes. Now, we have triangular meshes with maybe 30,000 triangles. In practice, we subsample them very coarsely. We maybe take 1,000. But what that means is that we don't really have in practice that the points go to corners of new triangles. We subsample too coarsely for that. We really go somewhere in the center. So if we went to the corners, then you would have that your map of labeling to which points you go would have ones between the A points and the B points. But in practice, we go somewhere in the center. And what that means is that you have that the point where A goes is a combination of the points of the triangle and where it gets. So you already have in your mapping something where you don't have an exact point-to-point correspondence. What that means is that you are looking at your surface and moving away from points. So when you look at your mapping from point to point, you also have a little move away from the new edge points, and so you have some motion on each surface. So now what we're going to do is think of this whole thing as a manifold of teeth, but every point on that manifold is really a tooth. So you have a fiber bundle. I mean, so your distance graph comes not just with a distance, but with a map from this surface to that surface. So, okay, so in a fiber bundle, what you have is a base manifold, like here the cylinder and the Möbius band. And at every point on the manifold, you have an additional structure. And these structures relate nicely to each other. And in this case, the structure is just an interval. And locally on the Möbius band, you don't know that you're not on a cylinder. I mean, locally you have a nice, so it's only when you start tying things together globally that you can see that just knowing the local structure of your base manifold and the fiber is not enough to characterize the whole manifold, since I have two different fiber bundles here. So that's the fiber bundle. And what you can then do is, if you have your fiber bundle, you can imagine, so at each point you have a fiber, you can imagine that diffusion has two components. It has a component where you walk on the manifold, but if you also have a mapping from the fiber to its other fiber, then you can lift that to via that mapping to a walk there. And you can do that all over. And what we have via the construction with our imperfect distances and so on is a way in which we can try to diffuse between the points on the base manifold as well as on the fiber. And the idea is then, can you, first of all, calculate with that in this whole framework, and can you try to disentangle the two diffusions? Because we're interested in the structure of the base manifold, not the fibers. Fibers we can see. So if you do diffusion map, this is typically what you do. You have your distances, you make it distances squared over epsilon because you're looking at very short time diffusion. You put a preconditioner in front. You actually make it so that you look at the diagonal elements if you sum and you invert that, and there are reasons for doing that, which I don't want to get into. Instead of doing that, each of these entries here will now be the whole surface. And each of these will be the mapping also for going from the I surface to the J surface with a little precision jump. And so what you then can try to do is find the horizontal component of those diffusion maps. So there's some technicality here. So if you have your diffusion maps, then you go from here to you want to do the horizontal diffusion map where you only work horizontally, and once you so you can try to disentangle these two, and we have some numerical results where he tried this out for the unit tangent circle, the unit circle on the tangent plane of a ball. So you take a ball, you take all the tangent planes, you take the circle, and that actually is a realization of SO3. And what you could do with that is you could then look at diffusion. You had two parameters to play with. One diffusion on the base manifold and one diffusion on the fibers. And depending on what you played with, you really saw, even if you made a lot of noise on your mappings, you saw the different spectral realizations that you would expect for the full SO3, for the SO2, for the fibers. I mean, they all came out very nicely. And all that is in a paper on the archive that I think you should all go visit as soon as you step out of this lecture. You look up Tingran Gao archive and you find it. Okay, let me show you applications on this teeth manifold. So one thing you could do with that is global registration, meaning we have a way now, I mean, after you've done that in this fiber bundle diffusion, you have a denoised version of the fiber that you get from putting all these fibers together. So you can use that in order to register. You can take a point on one of these, bring it to the fiber, see what then the nearest point is on this, and go back. And so in doing that, we have a better correspondence than what we found earlier with more primitive methods. And biologists were impressed by that. You can also do global registration. What you can do is you can try to register all the teeth together easily, and landmarking. So here what we did was we had all these teeth together, and they one of the teeth was then put into zones, and the zones were actually no, automatic landmarking. What we did was we took points on one tooth, and then we brought that back to the fiber. And then on the fiber, we looked at Voronoi regions, and then we mapped those to all the teeth together. And so instead of saying a landmark is a point, we said the landmark is a zone of influence. And we have all these zones of influence. And again, this is something the biologists liked. And then finally, the horizontal diffusion maps. Once you have a diffusion map, you can use them to make a distance. And in that distance is where Tingran, when he clustered, found in that distance the different species, the data points just cluster so nicely. And when they saw the nice separation between species, the biologists were really excited. They said, oh my God, maybe we are seeing the evolutionary and so on. But then when they looked at it, they said, it's really surprising because the different species, I mean, you see here green and blue should in a phylogenetic tree have been closer, and they're not. Blue is over here and green is there. And I mean, so it didn't work out. But fortunately, what happened is that they realized that in fact what they were seeing was that the frugivores were going together and the folivores were being and so on. So it was the function that was being found, which is not so surprising if you only look at teeth. I mean, how could you have imagined that you were going to see evolution? We don't do miracles. We look at the shape of these things. And so they laughed and they said, oh, well, form follows function, which is, of course, a big heresy in biology, because it's what Lamarck used to say. So we have these three things that Tingran's new approach really showed that so they now believe much more in us again. But this new insight, I mean, all this was possible because we took the mappings. And the new insight that the mappings are the fundamental and so on has made us reconsider how to start on this problem again. And again, we're going back to computer graphics. So we're working, as I speak, we're working on putting together bigger data sets where we have, again, like for Tingran's thesis, 10 specimens for each species that we look at, and we will have something like 20 different species, and which all will be landmarked, each one of them. Now, in computer graphics, in the meantime, some students of Jaron Lipman, with whom we're still working, who's now at the Weizmann Institute, have put together very nice ways of, if you have 20 correspondence points, of mapping one surface to another. The complete surface, not just the landmarks. And I thought first we'll show these mappings to our biologists, and they will not be happy with these mappings. They will say, oh, this mapping is not correct because that point should have been there and not there. But when they find such a point, we just add a new landmark based on their intuition, until we have enough landmarks that we have mappings that they like. And so far, actually, to my surprise, this hasn't happened. Every mapping we show them, they have liked. But the end result is that we want to have a whole collection of landmark stuff and the mappings between them. And then we will use that as a kind of higher level machine learning things. We will have surfaces and their geometry. We will forget about the landmarks, we'll just have the mappings. The mappings embody the biological knowledge of the biologists. And we will learn how to build mappings between surfaces from that very rich data set. And once we do that, we will then, of course, test it on other surfaces. And if we can do that, then we will have what we wanted. We didn't have to build these distance matrices. We had to build the mappings. And the mappings will give us richer interpretation anyway. And the mappings is what they really want. And that then will be, but so we're now at the start of that very interesting but different type of machine learning problem. And I'm going to stop here.
Thank you, Ingrid. So I think we have time for questions.
Sure.
The floor is open.
You said the visualization these interesting questions, and I was just curious if the biologist had an explanation. In this graph, and more so in the previous Diffusion mapping graph, of all of them, the blue dots are the least clustered. Did that prompt questions or answers from the biologist that you were dealing with?
Yeah, it's not as bad as it shows here because, I mean, if you look at it in 3D, I should have brought the MATLAB thing with me. It doesn't look as bad. But you're right. They were wondering about this point, and we don't really have a good explanation. I mean, it's not it's not really on the other side of the purple ones. I mean, but if I started moving that, then some of the other ones would have become less visible. So, but there was definitely an outlier in that species. This is an outlier too. But again, these were the only 5 species for which we had 10 specimens of 1. You see, it's a tedious thing to have to put these points on there. And they typically do just enough of them, if they have to do several within one species, that they get their p-value below the sacrosanct 0.05, and then they can publish. And that's why we really they want really an automatic method, because they have many more samples than they have the appetite or the resources to do all that manual landmarking for. And but so yeah, for the moment we need to, because we need to validate. But
more questions? Mark?
So clearly some IPAM programs are popular thing to do with sulci, with grooves in the cerebral cortex.
yes.
To try to match those. Have you thought about that?
Okay, yes and no. The sulci, the matching of the brain, the major sulci are clearly something that we all have in common. However, the individual folds of the brain are very individual. Even identical twins don't have the same. I mean, this study has been done have the same geometry of their folded brain. There have been studies that have tried to look at registration of brains based on functional data rather than geometry, which suggests that in fact one might get better results that way. So I'm not sure that doing geometry matching of brains is the most promising avenue. However, there are many other things than these surfaces. For instance, there are surface matching for surgical purposes. I mean, before doing a complicated surgery, typically now a geometrical imaging is done, and different surgeons or different experts talk together on how they will go about this. Now, once the patient is anesthetized and on the operating table, things look slightly different. I mean, it would be great to have an immediate remapping of that whole strategy on the actual concrete situation. So that's definitely a possible application. We're working with somebody who actually just started 2 weeks ago. So for the moment, we just are looking at data. We don't know yet what we can do. But with somebody at Duke who is as a mathematician working with surgeons on the nasal passages. There are versions of what a cleft palate is that in fact are not observed at birth because they don't have the exterior characteristics, but they do have the asymmetry inside, and that cause endless problems. And the way that gets surgically corrected, and the way they plan for that is they first model that whole passage, and they do that with CT, with CAT scans. But for small children, they don't want to use too many CAT scans. So they're trying to do it with MRI, which gives them less precision because these children don't sit still. And so in order to validate that they still have enough information, they want to use our methods. Before, in order to look at how much information they had in the nasal passage, they would model it and then look at a couple of sections and compare these geometric flat shapes. But we said we can look at the whole 3D shape, and they were very excited, and so we're going to look at that. So there are many other applications.
I was wondering that if your work leads to some generated models of how teeth get formed.
Okay, so we'd love to, so our work doesn't lead to that, but Jukka Jörnvall, together with a postdoc of his, Salazar, built a few years ago a model, and it made a Nature paper, in which they used understanding of genetic factors and growth models of teeth in order to, with variations in these few parameters, so they had a small number of parameters, and they produced teeth that looked very much like different teeth that are found among mammals. People were very excited, and so we were excited because we said, can we use your generative models in order to make many, because then we have no problem with sampling. And after much humming and hawing, we did get a copy of the MATLAB. The problem is, this is done in MATLAB, and when people program in MATLAB and they don't think of sharing their code ahead of time, they don't write it in a version that they feel comfortable sharing afterwards. And and it turned out that the program was not really generating surfaces. It was generating different clusters of things that they could interpret, but we need surfaces. And so I've been thinking about trying to wrap it in a surface. I mean, implicit functions theory, and to get a surface out of it. But Doug is hiring a postdoc who's going to go back to that model and build us a good model from the start that will generate surfaces.
So
and
the reason I asked is because I was talking to a dentist here, and they really, what I was told is that the proteins make this mesh. So in real life, it gets 3D printed.
Yes.
So the proteins make this mesh, and within this mesh, 3D mesh, the mineralization happens. Mm-hmm. So your work reminded me that in real life, it's 3D printed, so we generate the models.
And but so we'd love we'd love to have generated models for other reasons. We'd love to have a way of sampling that manifold much more densely. I mean, so that we could actually do things with it. I mean, we now depend on the teeth. Now they have zillions of teeth, but yes.
Come along the call. Go ahead.
Do you see any feedback to the motion capture, computer animation, Pixar world of this?
I haven't seen any, but for the computer graphics side, I depend on Yaron Lipman, and Yaron Lipman is one of the people who's completely embedded in the SIGGRAPH world. So I think whatever, I mean, for the moment, it's still things that they develop that help us. But if ideas, I'm this fall, I'm hiring one of his best students as a postdoc, so things will I'm sure get fed back if there is anything to feedback. But it's I mean, Yaron first when he came, and then now the student who's coming, they're just very excited to be working on something that's related to science and not to video games. And I mean, video games pays a lot of computer graphics, but.
Okay, one last question.
So, how does this compare to like other methods like facial recognition, emotional recognition, and these things that use neural networks and all that? We haven't a very different field, or
we haven't tried neural networks on these, but the biologists we have tried things that other distances between surfaces that exist, like Gromov-Hausdorff distances. They don't really work well for this at all, and they're very computationally much more heavy. Paradoxically, with our way of defining it into mappings, we will be now in a setting where we have parameters that we can actually try deep learning and with neural networks on. Before, we didn't really know how to even do it, but and we will try it.
All right, thanks very much, Ingrid, for inspiring us.